The following segment is intended for informational and educational purposes only and was written and developed by Bloodstream Media.
There is obviously a difference between the hemophilia A gene therapy product and the hemophilia B gene therapy product in data, efficacy, all of those words that we use. Tell me why. Why is there such a difference? Why are we seeing different outcomes?
Super good question. There's a lot to talk about, but I think, the biggest difference so far in the currently available gene therapies that we have and the things that are currently being studied in the sort of late phase clinical trials, the biggest difference between hemophilia A and hemophilia B gene therapy seems to be durability. In other words, how long do the factor levels stay up? So for factor 9, hemophilia B gene therapy, it seems that whatever response you get, it seems to be stable over time, at least out to 3 years or so. With the hemophilia A gene therapy, what we're seeing is the factor levels tend to fall off over time. They don't seem quite as stable, that's right now the major difference between the two. The reasons for that are still not completely understood. We talk a lot about part of this probably being related to the fact that the way we're currently doing gene therapy for hemophilia A and B is we're introducing the corrected copy of the gene into a cell called hepatocyte. It's a liver cell. The hepatocyte is where factor 9 is normally made, but it's not the cell that normally makes factor 8. So although other cells can make proteins, different cells that aren't the cell that normally makes a protein can be given the instructions to make a protein. When you ask a cell that doesn't normally make factor 8 to do so, it may not be as easy as just telling the cell to do it and giving it the tools. So there's some speculation that part of the problem between factor 8 and factor 9, or part of the difference, is that we're asking the cell that normally makes factor 9 to make it, and that goes okay. But we're asking a cell that doesn't normally make factor 8 to produce it, and it may not be quite as easy as we thought it would be. So there are probably other reasons as well, but that right now seems to be where a lot of the discussion is centered. We need to figure that out. I mean, why it seems to work differently for factor 8 and factor 9 is a really important question, and that's something that has to get sorted out to take this forward.
What's the cell that makes factor 8?
So factor 8 is normally made it's made in the liver, but it's in a different cell, not the hepatocyte. It's made in a cell called the sinusoidal endothelial cell. So the endothelium is the cells that line the blood vessels, and the sinusoids, which are little vascular spaces in the liver. That's where factor 8 is made. So a little bit different place than factor 9 is made. And both the currently available gene therapy treatments deliver the gene therapy to the hepatocyte, so into the organ, but not to the exact same cell type that normally makes factor 8, and that probably has a fair bit to do with why it looks a bit different.
What is by the way, can you say that cell name 3 times fast?
No, I can't. Hepatic sinusoidal endothelial cell. Yeah, that's a whole mouthful.
I'm so impressed you got that out. You've obviously said it before. But what would it take to get that, to administer into that cell?
Yeah, so the current platform that we're using for gene therapy, we're using something called adeno-associated virus, AAV. This is a form of a virus that's really good at delivering genetic material to a cell, and we can target these things to certain cell types, and targeting the hepatocytes is relatively straightforward. Targeting other cell types is possible, but a little harder. And so I think that's sort of the superficial answer to the question. I don't know that we're going to see this as the end game for gene therapy. I think it's becoming pretty clear that we may need to move past the current AAV platform, and we may be using other targets. We may be using other cell types to deliver gene therapy in the future. So there's a lot of work going on. There are other gene therapy studies going on right now using other cell types as the target, and so we'll see how that works out and how it compares to what we're currently doing. But I think it's pretty safe to assume what we're doing right now is gene therapy 1.0. We're going to have to make improvements on this platform to get the results that we want, and that might require moving beyond the AAV platform.
Another question, feel free to be candid as much as possible. As a physician, talking with patients in the clinic space, considering their treatment options, what are the questions or the concerns that hemophilia A patients are having versus hemophilia B patients?
Honestly, I think the concerns are kind of similar between the two patient groups. I don't know that the concerns are all that different. When I talk to my patients, the questions that we talk most about are, how well is this going to work in me, and how long is it going to last? And for factor VIII, obviously the how long it's going to last question is a bit bigger issue. But the first question is, how well is this going to work in me is applicable to both hemophilia A and B. And one of the challenges with the way we're currently doing gene therapy for hemophilia is that we can't predict how well it's going to work in a given person. The data that we have shows that average factor levels are, whatever they are, and that looks great. But it doesn't matter what average is; it matters what you are, as an individual, right? And the range of responses that we see is quite wide. So if you look at all the data for all the different gene therapy programs that are available and being studied, they all have the same issue in that there's a wide variability in how individual patients respond. So some people get factor levels that are normal, and some people's factor levels never get out of the single digits. And we don't really understand why that is, and we don't know how to predict how a given person is going to respond. So most of our conversations in the clinic with patients who are considering this are centered around those issues and really helping them understand that. Some still want to go forward and do it, and that's great, and we can support that. But you have to understand that piece of it before moving forward.
Once again, this segment has been brought to you by CSL Behring, which now has a first-of-its-kind hemophilia B treatment. Visit beyondhemb.com or download B Support wherever you get your apps for more information.